This lecture is over Chapter 21, the Digestive System, Functions, and Processes. Please make sure that you refer to your Chapter 21 study guide to see the learning objectives that goes along with this lecture and to see the material that you'll be responsible for. Let's begin with the anatomy system. So if we follow the track, we have first the oral cavity. We'd move into the esophagus, followed by the stomach. From the stomach, we would move into the small intestines. From the small intestines, we would then move to the large intestines, and then the rectum, go through the anal sphincter, and we're out. The glands that will be associated with the digestive system are going to be the salivary glands. We then have the pancreas and liver and gallbladder that would be associated with the small intestines. Each of these organs, if we look at the tissue layers that make them up, they share some common stratum or some common sections. The most inner layer that would be towards the lumen side, right, is called the mucosal layer. And in the mucosal layer, here we see this is the stomach, this would be the small intestines. This is the largest layer, and it's where we're going to have all of our secretory cells, and there's an effort to maximize surface area because this is where digestion and absorption is going to occur. So in the stomach, we have the rugae that create the ridges that increase surface area. In the small intestines, there's the villi that increase the surface area, and we have all of the cells that then secrete into these orifices. Below the mucosal layer, we have the submucosal layer. This is made up of connective tissue. We have our arteries and veins that are running through this, and we have our enteric nervous system, so the neurons that play a role in the digestive system run through the submucosal layer. Below the submucosal layer, we then have the muscularis externa, and this layer is where we have all the smooth muscle, so this is where the contraction force will be generated. And we also have enteric neurons in this layer as well. And then the most basal layer is the serosa, and the serosa really is just a continuation of the peritoneal membrane that holds the digestive system together and forms the mesentery that holds the digestive system within the abdominal cavity. The processes and functions of the digestive system are fourfold. They are secretion, in which we're moving material from the cells in the digestive system either into the lumen of the digestive tract or into the blood. Digestion, which is going to be the chemical and mechanical breakdown of food into absorbable units. We have absorption, which is now where we're taking material from the lumen and moving it into the ECF. And finally, the last process is that of motility, in which we're moving material through the GI tract, utilizing that smooth muscle contraction. And all of these four processes face challenges in the digestive system, specifically because of the nature of the processes that are going on and the fact that it's open to the external environment. So those challenges include avoiding autodigestion, right? As we digest biomolecules, we have to make sure that we don't digest ourselves. There's issues for both secretion and absorption that have to do with mass balance. How do you secrete something or absorb something against its concentration gradient? And with that, how do you ensure that you don't have fluid loss, that osmosis doesn't lead to fluid either being lost through the digestive tract in the form of diarrhea or in the reverse of vomiting? We can't have excess fluid loss. We need to retain that fluid. And finally, because we're open to the external environment, we need to be careful that we don't let in pathogens and we don't have a means to defend against those pathogens to ensure that we don't bring on a pathological condition. Now, we'll talk about secretion and absorption and and digestion in just a minute, but I want to talk first about motility. So motility is accomplished by the smooth muscle that makes up the tissues of the digestive system. And as you remember from smooth muscle discussions, there's two types of contractions that are possible with a smooth muscle. So we can have a tonic contraction in which we keep a sustained tension in that muscle, and then we have relaxation moments. Or we can have phasic contractions in which we're mostly relaxed, and then we have a contraction that doesn't last as long. So in the digestive system, those tonic contractions, they're maintained for long periods of time, and we primarily find them in our sphincters. So there's a number of sphincters that we have. There's an esophageal sphincter that prevents movement of food from the stomach up to up into the esophagus or out of the mouth into the oral cavity. We have the pyloric sphincter that prevents food from moving into the small intestines before it's ready, or from the small intestines moving back into the stomach. And so the point of those tonic contractions is to keep food matter from moving backwards in the system. The phasic ones, those are going to occur in really our stomach and small intestines, and the purpose of those is more for mixing and propelling food in a in a given direction. With those phasic contractions, we have two types of phasic contractions. We have what are called peristaltic contractions, in which the contraction force occurs behind the food matter. So when we chew up food and swallow it, we call it bolus. And so as we swallow it, you can think about this in the esophagus. We have a contraction behind the food which pushes it forward. Where the food is, the muscle is relaxed. And then it's followed by another contraction once that food is moved directly behind it, and it ensures that we have a one-way direction of movement. That's peristaltic. Segmental contractions, which we find in the small intestines, the direction isn't meant, or the function of it isn't a directional movement. We're not trying to move forward or the food forward into the system. Rather, we're trying to mix it. And so we have these contractions that squeeze and move forward in both ways, that turn it over and mix it to ensure that the digestive enzymes come in contact with everything, as well as that the bolus comes in contact with the epithelial cells for absorption. Now, these functions of digestion, absorption, secretion, and motility are all regulated by what we call long reflexes and short reflexes. Long reflexes we've been talking about in a lot of our other systems. These are directed by the central nervous system. They originate outside the GI tract, and they typically are either hormonal or neural in how they originate. Short reflexes, these ones actually originate inside what we call the enteric nervous system. So inside the digestive system, there's a whole network of neurons that operate outside of the central nervous system, sometimes without even any interaction with the central nervous system. They are their own integrating center. You can kind of think about it like we have a little mini brain in our gut that directs a lot of the actions that go on in our digestive system. And so what we see is that from the central nervous system, we have input that comes through our sympathetic and parasympathetic neurons. And those tend to have actions either on the enteric neurons themselves or sometimes on the smooth muscle. And they can direct changes in motility, changes in secretion, enzymatic release, those types of things that can be controlled. Likewise, we can also have endocrine hormones that are secreted that can direct some of these things as well. So like insulin and glucagon play a role in glucose uptake, and those can be directed by the central nervous system as well. So everything in gray can be directed by that. However, we can also have these internal directions. So they can come from secretions that occur from the cells inside as well. They can then have an effect on the enteric nervous system. The peptides, we can have an effect on the smooth muscle. So there's a lot of coordinated regulation that occurs, and it's not just entirely from the central nervous system. A lot of it comes from inside the GI tract itself. I love this figure in this chapter. So this is figure 21.6, I believe. And these takes the 4 functions of the digestive system, and it outlines them for really kind of the 4 main parts of the digestive system: the oral cavity, the stomach, small intestine, and large intestine. So we're going to go through these really quick. In the oral cavity, as far as secretion goes, the main secretions that are occurring are saliva. We'll talk a little bit more in detail what's composed in that saliva, but we have some amylase and we have some isotonic sodium chloride, right? As far as digestion goes, the amylase found in saliva starts to digest some of our carbohydrates. It breaks down the starches and starts to make them into disaccharides. There is no absorption that occurs in the oral cavity, and the motility is accomplished through chewing and swallowing. Once we've swallowed, that food then enters into the stomach, and here we start having lots of secretions. So we'll have secretions in the form of gastric acid from the parietal cells. We'll have pepsinogen and gastric lipase that get secreted from chief cells in the stomach. Mucus and bicarbonate get secreted by surface mucosal cells. And then the digestional hormones gastrin and histamine will be secreted by G cells and our ECL cells in the stomach. The digestion that occurs in the stomach is going to be protein. So pepsinogen gets activated by our gastric lipase, and we digest. It becomes its active form of pepsin that helps break down our proteins. Fats, there's a little bit of digestion in the stomach that occurs with fats, but it's less than 1/3 of the total fat digestion would occur in the stomach. As far as absorption goes in the stomach, the only compounds that are absorbed are those that are lipid soluble, and that's substances like aspirin, alcohol. None of our biomolecules like that we think of, the carbohydrates, amino acids, those things aren't absorbed. And the motility is primarily peristaltic. When we move to the small intestines, now again we have a lot of secretions. There's lots of digestive enzymes. They get secreted, tons of what we call zymogens. We have mucus that gets secreted. A lot of gastric hormones are now secreted. So CCK, secretin, GIP, GLP-1, those would get secreted. Bicarbonate and bile would be secreted by the pancreas and the liver into these regions. Digestion, the small intestines is where almost all of our digestion occurs. Almost all the digestion and absorption occurs. So our polypeptides, our carbohydrates, our fats, our nucleic acids will be broken down in the small intestines, and they will be absorbed as well as amino acids, small peptides, monosaccharides, fatty acids, monoglycerides, cholesterol, our nitrogenous bases from our nucleic acids. Water gets absorbed, ions gets absorbed, minerals get absorbed, and vitamins get absorbed. And the primary means of motility is going to be segmentation. There's a little bit of peristalsis to move things forward, but most of it's going to be segmentation. Finally, in the large intestines, the only secretion is going to be mucus. There is no real digestion. There's some minuscule digestion of small organic molecules that are created by bacteria in the large intestines, but it's minimal. We do absorb a lot of water. There's some ions that'll be absorbed, some vitamins. And motility is going to be segmentation for mixing, and then mass movement of where we just kind of pack in the colon. As new stuff comes in, it pushes the others out. So make sure you know this figure, that you can identify these things and kind of go through that. Likewise, this table 21.1 is going to be important, not so much in this chapter's discussion, but certainly in the next chapter, we'll talk about these GI hormones. So stomach, we'll talk about this today, gastrin, and in the intestines, these ones we'll talk a lot about next time. For the rest of this lecture, what I'm going to do is I'm going to go through the digestion. We're not going to talk all that much about absorption. The book goes into a lot of absorption, the mechanics of it. We've spent a lot of this semester talking about absorption in other ways, so we're just kind of brush past that a little bit. But digestion, I want to hit a few points, and we'll break it up into the 3 phases of digestion. So the first is what we call the cephalic, that takes place in the oral cavity. And the digestion here is going to be chemical and mechanical in nature. It's the chemical digestion will occur by the saliva that is being secreted from the salivary glands. The purpose of that saliva is to lubricate the food so we can create a bolus and swallow. There also is some amylase and a little bit of lipase in there to help with some fat digestion in the stomach. The saliva also has a protective function in that it can remove out some of the bacteria and kill some bacteria. And the mechanical digestion occurs by mastication, which is chewing, and that's going to break up the particles, the food, into smaller, more manageable particles that can be swallowed. And that's followed by swallowing, which is what physiologically we term deglutition. And the way deglutition works is once you've chewed your food, you create a bolus and you push it against the soft palate in the back of the mouth. And what that does is it's going to trigger the closing of the glottis to ensure that we don't put food into the trachea and then choke on it. And it's also going to relax the esophageal sphincter. So that's what we see. The glottis closes, the esophageal sphincter opens, and then peristaltic contractions in the esophagus will move the bolus, along with gravity, into the stomach. Once we're in the stomach, we enter phase 2 of digestion, the gastric phase. And the purpose of the stomach is it serves 3 main purposes. The first being storage. It's really important that food is regulated as to when it passes into the small intestines, because the small intestines is where most of the digestion is going to occur, and it's where most of the absorption is. And so we don't want to be constantly pushing food into the small intestines if we haven't already digested and absorbed the food that's in there. So it serves as a storage container. It also serves as a defense mechanism. We protect the body by destroying a lot of the bacteria and other pathogens that may have been swallowed with our food. By the gastric acid that's in the stomach. And then it serves a function of digestion, of mechanically breaking down food into what we call chyme, that is a kind of a soup of digestive enzymes and mashed up food. There's a lot of secretion that occurs in the stomach, and I want you to be familiar with these secretions. So this is a great table from the book that kind of goes over that. We have cells that are found in the mucosal epithelial that secrete mucus and bicarbonate. And the function of that mucus and bicarbonate is going to be to protect the stomach. It forms a physical barrier between the lumen of the stomach and the epithelium. And the bicarbonate acts as a buffer, so as we secrete gastric acid into the stomach, we don't eat away the cells of the stomach lining. Right? When you develop a gastric ulcer in your stomach, it's usually because you're producing too much gastric acid, and this physical barrier isn't being maintained. Okay, we then have parietal cells, and the parietal cells produce the gastric acid that is important, one, to lower the pH in the stomach to kill bacteria. Lowering the pH also helps break apart some of the peptide bonds. And it also activates the digestive enzyme in the stomach of pepsin. So it's important for that. It gets triggered by acetylcholine, which gets released by our parasympathetic and our enteric neurons. And it can also be triggered by the binding of gastrin or histamine to receptors on the parietal cells. Histamine is secreted by our ECL cells. Histamine, again, it triggers the release of gastric acid. Chief cells, those are the ones that produce the digestive enzymes. So they're the chief digester cells. They produce and secrete pepsinogen. Once it gets activated by gastric acid, it'll be pepsin. And it secretes gastric lipase. And again, triggered by acetylcholine, and also acid, pH, will trigger the release of this. Lastly, we have D cells and G cells. So D cells produce a hormone, a gastric hormone, a digestive hormone called somatostatin. And this can bind to receptors in the parietal cells to actually stop gastric acid secretion. It says, look, we have too much acid. We don't need any more. When you take antacids, some antacids are just, acting as a buffer like bicarbonate to try to reduce that down. Some of the pills that you take, though, act to try to increase the somatostatin that gets released. We also have then the G cells that secrete the hormone gastrin, which again binds to the parietal cells that trigger the release of gastric acid. And what triggers the release of gastrin is acid. So just to kind of summarize, we have this regulation that's kind of going on inside the stomach. So as we bring in food, as amino acids and peptides enter into the stomach, those trigger the enteric sensory neurons to say, hey, look, we're filling up. They're stretch receptors. They recognize that. They go, we've got food in here. And they trigger the G cells to start producing gastrin. So as that gastrin gets produced, that triggers the parietal cells to start producing the gastric acid. So we start producing gastric acid. That lowers the pH, which then can have a negative feedback loop on our D cells to say, hey, look, it's getting pretty acidic. Let's create some somatostatin that kind of stops the parietal cell from producing quite as much. Likewise, that acid is going to trigger both enteric sensory neurons to then trigger chief cells to say, hey, let's start producing pepsinogen. And pepsinogen gets converted and activated by the gastric acid into pepsin so we can start digesting. So we see how this is all going. Additionally, the central nervous system can also play a role through parasympathetic neurons. Once we move out of the stomach, we then go into the intestines, and we enter the third and final stage, the intestinal phase of digestion. In the small intestines, there is going to be secretions that come from the pancreas and the liver. The pancreas secretes insulin and glucagon, which we'll talk about in the next chapter, that play a role in glucose uptake. But there's an exocrine secretion that goes directly into the small intestine, in which we put in both digestive enzymes that we call zymogens. Okay, zymogens will then be activated, usually by trypsin in the small intestine, and they turn into the enzymes that will break down our carbohydrates, our proteins, our lipids, our nucleic acids, and we also secrete bicarbonate. And the reason why the pancreas is secreting bicarbonate directly into the duodenum is there's a lot of gastric acid spilling in from the stomach when we move food in, and we need to neutralize that so we don't damage the intestine. So we secrete a lot of bicarbonate. The liver, its function is to produce bile. That bile will then be stored in the gallbladder and then be secreted into the small intestines. And the purpose of bile is to help emulsify fats. So fats aren't water soluble. You've seen this when you have an oil and water. It doesn't mix. If you want to, part of digestion is making things into small particles so you can increase the surface area for the digestive enzymes to work on it. And so in order for that to happen with fats, we need to break it up. We need to emulsify it. So just like putting soap into some oil and water causes the oil particles to kind of like make little globules, and you have all these little small bubbles. Same thing happens. That's what the bile salts do. It emulsifies it, turns them into really small micelles that then can have the digestive enzymes work on them, and we can transport them and absorb them out. In the digestion that then takes place, so all those zymogens are brought in, they're activated, and we then start to break down all of our biomolecules. So carbohydrates are going to be broken down into monosaccharides to be absorbed. Proteins get broken down into small peptides by our endopepsidases and our exopepsidases. So an endo breaks in the middle. That's like pepsin in the stomach. Exos break towards the ends. And those are the ones that we bring into the small intestines. Our lipids, we talked about the bile salts that help them be absorbed. Nucleic acids, they get broken down into nitrogenous bases and monosaccharides to be absorbed. And additionally, we absorb our vitamins and minerals in here. So lipid-soluble vitamins get absorbed with our fats. If it's water-soluble, they're going to need to be mediated, transported out. And one of those is vitamin B12. If we go back here, you'll see the parietal cells produce a second secretion called intrinsic factor. Intrinsic factor is needed to bind to vitamin B12 so that we can absorb it in the small intestines. We also absorb our minerals like calcium, iron, sodium, usually through active transport. And this slide, again, is just to kind of summarize, there's a lot of interaction going on. So I told you the stomach acts as a storage area because most of the digestion and absorption is occurring in the small intestines. And so we have lots of feedback loops. That's part of the reason why the small intestines produces so many digestive hormones, is we have these feedback mechanisms that slow the stomach down, both in its acid secretion and in its motility. To say, like, look, let's not move stuff. We need to wait. As we start bringing things into the small intestines, we start producing some of these hormones, things like secretin, CCK, right? And those all have these negative feedback. So secretin is important to stopping the acid from coming in. It has it triggers the pancreas to say, hey, let's start secreting that bicarbonate. That bicarbonate's important to neutralize the acid so that the small intestines doesn't get hurt. Additionally, it has a negative feedback loop on those parietal cells to say, hey, don't produce as much gastric acid right now. Like the small intestines is working. You need to store some food. The last part of the intestinal phase is going to occur in the large intestine. So we don't really have much digestion that occurs here. The only digestion would occur from byproducts that are being produced by bacteria. But we are concentrating our waste for excretion here. And so this large intestines is pulling a lot of water out, concentrating the waste. That movement, the bolus is moved forward until once it reaches the colon, we use mass action. When the colon is full, that triggers defecation, and there's a reflex pathway that releases the anal sphincter. It relaxes, and then we can use a peristaltic contraction and gravity to release and defecate. Last part is just an overview of the immune functions of the GI tract. I talked about how important that gastric acid is to killing bacteria in the stomach. Additionally, we have cells called M cells that are found all throughout the digestive tract that produce compounds called cytokines. And these cytokines are a signal molecule that trigger the inflammatory response, which brings immune cells to those regions. So we have M cells that basically are bringing immune cells into into the GI tract to take care of potential infectious agents. Additionally, we have things like the vomiting reflex to remove out poisonous substances that the body, usually it's linked to taste, where the body recognizes this is something that's not good. We've adapted over evolution a lot of these reflexes of knowing this is not something good. Or you get severe cramping that tells it like this is not good, and that triggers the vomiting reflex, and we release that out. And there's a lot of new evidence that shows that the bacteria that we actually have in our gut, called the microbiome, that it plays a large role in our overall immune function and health, and they can also secrete compounds that protect the body from pathogens as well.